Assalamu alaikum wa rahmatullahi wa barakatuh. Hello everybody, how are you? Hope you are all well. I am Dr. Dua Karim Metwalli, lecturer of histology. In this video, we are going to discuss the first part of the integumentary system, skin and its appendages. What are our ILOs for this lecture? By the end of this lecture, you should be able to list types of skin, list types of cells of the epidermis of the skin, describe the structure and the correlated functions of keratinocytes, name the 5 layers of the epidermis, explain pigmentation of the skin, list types of non-keratinocytes, describe the structure and correlated functions of melanocytes, differentiate between the melanocytes and melanophores, understand the structure and function of Langerhans cells, understand the structure of Merkel cells. What is the importance of skin? What are the functions of skin? The skin covers our body surface, protect the deep internal organs, protect our bodies against ultraviolet rays by its content of melanin. Sensation, as it contains receptors in epidermis like Merkel cells and free nerve endings, receptors in dermis like Meissner corpuscles, Bassinian corpuscles, cold receptors. Also, it is important for excretion of waste products in sweat. Also important for thermoregulation by sweat, blood vessels, fat in hypodermis. It also has immune function, prevent the entry of the organisms by its content of keratin granules, lamellar granules, also contains macrophage in dermis, antigen-presenting cells like Langerhans cells. It also has antibacterial effect due its content of the sebum and sweat. Also involved in synthesis of vitamin D3. It also responsible for the pigmentation of the skin because its content of melanin pigment may contain carotene pigment, hemoglobin in blood, and the presence of the numerous blood vessels. The skin is the largest organ in the body, constituting about 16% of the total body weight. It consists of 2 layers of different origins. The epidermis, which is ectodermal in origin, and the dermis, which is mesodermal in origin. The skin rests on subcutaneous adipose connective tissue layer called the hypodermis, which allows skin to move freely on the deeper structures. The hypodermis is not a part of the skin. The epidermis is the surface or superficial layer of the skin. It is formed of stratified squamous keratinized epithelium, so it is a vascular structure. It is ectodermal in origin, increases in thickness over palms and soles. The epidermis is formed of stratified squamous keratinized epithelium. It forms invaginations into the underlying dermis. These invaginations called epidermal ridges. The epidermis is separated from the underlying dermis by the basement membrane. This is the dermis, and this is the epidermis. the epidermis separated from the underlying dermis by the basement membrane of these cells. There are four cell types in the epidermis: keratinocytes, melanocytes, Langerhans cells, and Merkel cells. Another classification may be keratinocytes and non-keratinocytes. Keratinocytes, cells found in all layers of the epidermis, and non-keratinocytes, which include the melanocytes, Langerhans cells, and Merkel cells. Let's start with the keratinocytes, the first type of the cells. Keratinocytes are the most dominant cells of the epidermis. It can divide, migrate up, and undergo keratinization to form a protective layer for the skin, which is the keratin. These are the 5 layers of the epidermis. Keratinocytes present in all these layers. They have the ability to divide and migrate up to form a protective layer of the skin, which is the keratin. Keratinocytes are arranged in 5 layers, which are the 5 layers of the epidermis: stratum basal, the basal layer; stratum spinosum, and also called Brickell cell layer; stratum granulosum or granular cell layer; stratum lucidum, clear cell layer; and finally stratum corneum or horny cell layer. The stratum basal or the basal cell layer, it is the deepest layer of the epidermis. It is formed of a single row of columnar or cuboidal cells resting on a basement membrane. The cells have basophilic cytoplasm, and they are attached to one another by desmosomes. The cells are also attached to the underlying basement membrane by hemidesmosomes. In a light microscope, the cells show mitotic figures as they are responsible for regeneration of the epidermis and serve as stem cells for the epidermis. By electron microscope, cells show or contain intermediate filaments called tonofilaments or keratin filaments. So the cells of the stratum basal or basal cell layer with the light microscope appear as a single layer of columnar cells, basophilic with basophilic cytoplasm and basal oval nuclei, and show mitotic figures. And these cells rest on basement membrane. With electron microscope, the cells bind to each other by desmosomes and to the underlying basement membrane by hemidesmosomes. The cells contain free ribosomes, intermediate filaments, or tonofilaments or keratin filaments, and contains also melanin granules. Next layer, which is stratum spinosum or Brickell cell layer, It is the sixth layer of the epidermis formed of multiple rows of polyhedral or polygonal cells with central rounded vesicular nuclei. Spiny projections extend from these cells and interdigitate with projections from other cells in the same layer. Cells are held together with desmosomes at the tips of the spiny projections. This spines is responsible for the naming of this layer, stratum spinosum or brachial cell layer. The keratin intermediate filaments or tonofilaments increase as the cells divide and move upwards. These filaments assemble into bundles called tonofibrils that terminate at the desmosomes. The deepest cells in this layer are mitotically active, like cells in the basal cell layer. By the light microscope, the layer, the brachial cell layer, appear as multiple layers of polygonal or polyhedral cells with rounded vesicular central nuclei. The basal cells of this layer show mitotic figures. By electron microscope, these cells adhere to each other by desmosomes and contain keratin intermediate filaments or the tonofilaments, melanin granules, and lamellar granules that start to appear in this layer. And these lamellar granules are membrane-bounded granules. The basal cell layer and the basal part of the stratum basal, the nuclei of cells of these parts show mitotic figures, as the cells are responsible for regeneration of the epidermis, and hence the name germinativum. Human skin or the epidermis is renewed every 15 to 30 days. Again, these parts, the basal cell layer and the basal part of stratum spinosum. The cells the nuclei of these cells show mitotic activity and are responsible for the turnover of epidermal keratinocytes. And these both parts called Malpighian cell layer or stratum germinativum. The stratum granulosum or granular cell layer by the light microscope, it consists of 3 to 5 layers of flattened cells with central oval nuclei, and the cytoplasm, as we see in this picture, contains deep basophilic granules. The cells of this layer are undergoing the terminal differentiation of keratinization, as the cells at the basal layers start to contain the keratin filaments, and the keratin filaments, as the cells go upwards, increase and becomes more mature. Keratin filaments increase in the upper layers and become more mature. So the cells in the stratum granulosum granular cell layer undergoing the terminal differentiation of this keratin intermediate filaments. By electron microscope, the cells adhere to each other by desmosomes and contain the intermediate filaments, tonofilaments, and two types of granules, keratohyaline granules and lamellar granules. The keratohyaline granules are non-membrane bounded basophilic masses, contain proteins associated with tonofibrils and link them into larger structures. The lamellar granules are small membranous structures. They are membrane-bounded granules, contain lipid and lipoproteins. As they librate or exocytose into the intercellular space, they act as cementing substance, adhering the cells to each other, producing impermeable lipid-rich layer around the cells, forming barrier against water loss, act as waterproof for the skin against penetration by foreign materials. The stratum lucidum, or the clear cell layer, it is found only in thick skin. It is a thin, clear or homogeneous line, which is formed of non-nucleated flattened cells without any organelles, but attached together by desmosomes. The cytoplasm consists mainly of filaments, still contain the intermediate filaments, tonofilaments, keratin tonofilaments, and also contain Leyden granules derived from keratohyaline granules. Again, this layer present only in thick skin. It appears as a thin translucent layer of flattened eosinophilic cells, like the nuclei and organelles as they lost, and still contain keratin filaments, and the cells are held together by desmosomes. The stratum corneum, or horny cell layer, it is the most superficial layer, consists of several layers of acidophilic horny scales or horny cells, which have neither nuclei nor organelles. It is formed of about 15 to 20 layers of squamous keratinized cells filled with keratin filaments. These cells are filled with keratin tonofilaments embedded in amorphous matrix. These scales or these cells, horny cells or scales, are firmly adherent to each other by remnants of desmosomes. However, these scales are continuously shed off from the surface and become replaced by new ones from the deeper cells by mitosis. Again, the stratum corneum, or horny cell layer, in this layer, the scales are firmly adherent to each other by remnants of desmosomes, and the scales are continuously shed off from the surface and become replaced by new ones from the deeper cells by mitosis. During the keratinization process, the hydrolytic enzymes liberated during this process disrupt the nucleus and organelles, and they disappear as the cells fill with keratin. Okay, let's briefly summarize the layers of the epidermis. The epidermis formed of five layers with keratinocytes. The first layer, base, at the base, stratum basal or basal cell layer, formed of a single layer of columnar cells resting on basement membrane with basal oval nuclei. And the cells show multitic figures as they are responsible for regeneration of the epidermis. As human skin or epidermis is renewed every 15 to 30 days. The cytoplasm contains intermediate filament stonofilaments, melanin granules, and the cells attached to each other by desmosomes, and the cells bind to the underlying basement membrane by hemidesmosomes. The second layer, brachial cell layer or stratum spinosum, formed of multiple layers. In light microscopes, the cells appear polygonal or polyhedral with central vesicular rounded nuclei. The basal cells of this layer show mitotic figures like the basal cells. And so these two parts called stratum germinativum or Malpighian cell layer. The cells by electron microscope adhere to each other by desmosomes and contain melanin granules and intermediate filament stonofilaments, and start to show in the upper cells lamellar granules. Lamellar granules, which are membrane-bounded granules, contain lipid and lipoprotein. When liberated into the intercellular space, they act as a cementing substance, forming a barrier, adhering the cells to each other, prevent evaporation of the water, and act as waterproof for the skin. The next layer, which is stratum granulosum. Stratum granulosum formed of 3 to 5 layers of flattened cells with central oval nuclei. Contains basophilic granules. By EM, these cells adhere to each other by desmosomes, and contains intermediate filament stonofilaments and two types of granules, lamellar granules and keratohyaline granules. Stratum lucidum, which is present only in thick skin, appear as a thin, clear, homogeneous layer formed of flat cells with no nuclei or organelles, and cells attached by desmosomes, and still contain intermediate filaments, keratin tonofilaments, and also contain elidine granules derived from the keratohyaline granules. The stratum corneum, as we see here, it is the most superficial layer, as we mentioned, formed of several layers, about 15 to 20 layers of flattened non-nucleated keratinized scales, horny scales or horny cells, formed of keratin tonofilaments embedded in amorphous matrix. The cells still attached by remnant of desmosomes, and they are continuously shedded off from the surface and become replaced by new ones from the deeper cells by mitosis. Okay, let's now discuss the non-keratinocytes. The non-keratinocytes of the epidermis are the melanocytes, Langerhans cells, and Merkel cells. Melanocytes are ectodermal in origin from the neural crest. They are located between cells of stratum basal and in hair follicles. By the light microscope, the cells appear rounded with long cytoplasmic processes that branch into the epidermis between the keratinocytes. They have single rounded central nuclei, and the cells give positive Dopa reaction due to the presence of tyrosinase enzyme. By the electron microscope, these cells are not attached to the keratinocytes by desmosomes, but attached to the underlying basement membrane by hemidesmosomes. The cytoplasm is rich in mitochondria, rough endoplasmic reticulum, Golgi apparatus, and contains membrane-bounded melanin granules in different stages of development. These granules called melanosomes. These melanosomes contain tyrosinase enzyme, which converts the tyrosine to DOPA, then melanin. So melanocytes are DOPA positive cells contain tyrosinase enzyme. The mature granules, mature melanosomes, are ellipsoid in shape and are present at the tips of the cytoplasmic processes. As we mentioned, the cells attached to the underlying basal lamina by hemidesmosomes, but not attached to the neighboring keratinocytes. Melanocytes are responsible for synthesis of melanin. Let's now know how the melanin is formed. The tyrosinase enzyme is a protein. It is synthesized in the rough endoplasmic reticulum and go to the Golgi apparatus packed in membrane-bounded vesicles. Tyrosinase enzyme converts tyrosine into DOPA, which polymerize into melanin inside the vesicles, which is called melanosomes. So the cells is DOPA positive. The melanocytes are DOPA positive cells. The melanosomes, as they mature, they are transported to the tips of the cytoplasmic extensions. The neighboring keratinocytes phagocytose the tips of the cytoplasmic extensions containing the melanin, and this is called the cytokine secretion. So, the keratinocytes get the melanin granules, obtain the melanin granules from the melanocytes by cytokine secretion. Keratinocytes can't synthesize the melanin granules. So keratinocytes are DOPA negative cells. They contain no tyrosinase enzyme. The only cells that can produce and synthesize the melanin granules are melanocytes, as they contain tyrosinase enzyme, which convert the tyrosine to DOPA, then melanin. and become membrane bounded into vesicles called melanosomes, and mature melanosomes are transported to the tips of the cytoplasmic extensions, and then keratinocytes phagocytose these tips containing the melanin. And this help to scatter and absorb ultraviolet radiation of sunlight, which damage DNA, producing mutagenic effects. The sunlight increases production of melanin and changes also chemical characteristics of melanin. Although melanocytes produce melanosomes, the keratinocytes contain more of this pigment than melanocytes. What are the epidermal melanin units? It represents one melanocyte with the keratinocytes into which it transfers the melanosomes. The density of these units is similar in all individuals. What determines the color of the skin? Black, white, albino. It is related to the rate of the production of the melanin granules and their accumulated amount, its amount in the keratinocytes. This is what determines the color of the skin. The rate of production of melanin granules and its amount in the keratinocytes. We can face many clinical conditions related to the skin and its pigmentation, like albinism, vitiligo. Albinism is a congenital disorder which is characterized by skin hypopigmentation. And this hypopigmentation is due to defects in tyrosinase production. The cells can't produce the tyrosinase enzyme. There is a problem in or a defect in production of the tyrosinase enzyme, so the melanin granules can't be produced. Another condition, it is vitiligo, which is an acquired condition characterized by patches of skin depigmentation. As we see, patches of skin depigmentation due to loss of melanocytes. The exact cause is not clear. The cause of this condition is not clear. Okay, let's now know what is the difference between melanocytes and melanophores. Melanocytes, as we mentioned, they are specialized cells found in the epidermis between the basal cell layers and have many cytoplasmic processes. They are ectodermal in origin, responsible for the synthesis and production of melanin. They attached to the underlying basement membrane by hemidesmosomes, and these cells form the tyrosinase enzyme. So these cells are DOPA positive cells, as they through the tyrosinase enzyme are able to convert the tyrosine to DOPA, then melanin. So the presence of DOPA in these cells make these cells positive, have a positive DOPA reaction. While melanophores, like pigment cells, they are connective tissue cells. They are connective tissue cells. They are mesodermal in origin. Phagocytose the melanin from formed by melanocytes. only phagocytose the melanin formed by melanocytes, and they are present in the dermis of the skin. These cells are DOPA negative cells, as they have no tyrosinase enzyme. They can't produce tyrosinase enzyme, so these cells are DOPA negative. The second cell of non-keratinocytes is Langerhans cells. They originate in bone marrow and migrate via bloodstream to the skin. They are located mostly between the cells of stratum spinosum, but not connected to them by desmosomes. By the light microscope, these cells have long cytoplasmic processes, and these processes extending between the keratinocytes of all layers. Their cytoplasm is pale, and they have dense nuclei. By the electron microscope, the cytoplasm of Langerhans cells contains well-developed Golgi apparatus with spares rough endoplasmic reticulum. The rough endoplasmic reticulum is not very prominent. The cytoplasm also contain mitochondria, abundant lysosomes, and multivesicular bodies. It also contains membrane-bounded granules, racket-shaped, called Purbeck granules. As we see, racket-shaped, membrane-bounded granules called Purbeck granules. Purbeck's granules. Langerhans cells protect the skin from foreign invasion and play a role in the immune system. As they act as the macrophage of the skin. They are antigen-presenting cells. They can bind, process, and present the antigen to the T lymphocytes, stimulating them. They also play a role in contact allergic reaction. The last cell of the non-keratinocytes is Merkel cells. They are also called epithelial tactile cells, few in number. They are present in the basal cell layer of the epidermis and are most numerous in thick skin. They are located between cells of stratum basal, joined to them by desmosomes, and they have dense core neurosecretory granules. The cells are slightly larger than the neighboring cells. They interdigitate and are attached to the keratinocytes or epidermal cells by desmosomes. The nucleus is single and central with indentation. The cytoplasm is pale, containing dense granules, as we mentioned, that it is characterized by dense core neurosecretory granules. They are associated with Merkel's disc, this disc, to which they are closely applied. The sensory nerve fiber loses its myelin sheath as it reaches the epidermis, and the naked nerve fiber penetrates the basement membrane to terminate as terminal disc-like expansion under the Merkel tactile cell. Again, Merkel cells present in stratum basal between the cells of the basal cell layer. They are branched cells with central indented nucleus. This nucleus show indentation. attached to the keratinocytes by desmosomes. The sensory nerve fiber loses its myelin as it approaches Merkel cell and form disc-like expansion. Merkel's cells are modified sensory receptor cells. They are regarded as mechanoreceptors for touch sensation. Let's summarize our lesson for today. The skin consists of two layers, epidermis and dermis, and the hypodermis is not a part of the skin. The epidermis is formed of stratified squamous keratinized epithelium. There are four cell types in the dermis: keratinocytes, melanocytes, Langerhans cells, and finally Merkel cells. Another classification: keratinocytes and non-keratinocytes. The keratinocytes are arranged in five layers: stratum basal or basal cell layer, stratum spinosum or brachial cell layer, stratum granulosum or granular cell layer, stratum lucidum or clear cell layer, and finally stratum corneum horny cell layer. The non-keratinocytes are melanocytes, Langerhans cells, and finally Merkel cells. Thank you so much for your listening.